Could I have everybody take their seat, please? Thank you. Well, good afternoon. Welcome to the Reilley Symposium. My name is Henry White, and I'm presiding over this afternoon's talks. I want to welcome everybody. We have a great lineup of presentations this afternoon, and we also have an opportunity to acknowledge the research contributions of the Reilley awardee and the Society of Electroanalytical Chemistry Young Investigator awardee. I thought before we start, I'd just like to mention for the youngsters here who Charles N. Reilley was. He was a professor of analytical chemistry at the University of North Carolina for a long time. He made many contributions to the field of analytical chemistry, specifically in the area of electrochemistry, and it's really a pleasure to get together every year and to remember and honor his contributions to this field. So let me proceed with the presentation here of the Reilley Award. This year, the 2013 Reilley Award goes to my friend, Andy Ewing. And Andy, if you'll come up here, let me tell you a few things about Andy. Andy is currently the chair of analytical chemistry at Chalmers University in Sweden. He's also the director of the joint initiative for bioanalytical chemistry between Chalmers and Gothenburg University. For those of us who've known him for a long time, we still remember Andy as a Nittany Lion from Penn State, where he was on the faculty for 25 or 26 years. And Andy has made many contributions to the fields of electrochemistry and bioanalytical chemistry. He has pioneered quantitative electrochemical measurements of single cells, in particular exocytosis from single cells and the use of capillary electrophoresis, using electrochemical detection to monitor the contents and the mechanism of chemical reactions that occur inside of single cells. He was the first, I believe, to measure neurotransmitter release from intact neurons. Many contributions. I have lots of personal stories, but I said I try to be a little bit distinguished this afternoon. I'm going to leave that to Andy's PhD advisor, Mark Whiteman. Andy has a number of connections to Charlie Reilley through Mark Whiteman, who was a graduate of UNC, and through his postdoctoral advisor, Royce Murray, who's in the audience. So without further ado, let me present the the 2013 Charles Reilley Award to Professor Andy Ewing. Andy, and I believe we're going to have the president of the Society of Electroanalytical Chemistry come up and make the joint presentation. I'm going to let you present it to him.
You have to turn around.
Yes, ma'am.
Okay, so this says the 2013 Charles N. Reilley Award presented to Andy G. Ewing to recognize his outstanding contributions to electroanalytical chemistry, sponsored by BAS 2003. Congratulations.
Thank you very much.
Congratulations, Andy. Thank you. Pittcon requires me to make two other announcements. These are required. No photography and no smoking. So with that, Andy, congratulations.
Thanks, Henry. And thank you for leaving out all those other comments. But you know I go next, so I guess that's part of it. And I think it's pretty funny that they tell you no photography, because this is being videotaped. Okay, so what I want to do is talk a little bit about three different areas of science that we do in my group. And so you have the title up here of my talk, and you can read it as well as I can. And yeah, you better start that. No, 55. We'll give you Mark 15 minutes, right? Okay. Okay, so, and we have, I'm going to talk about 3 different things today. I'm going to talk about electrochemistry in the brains of fruit flies. And I put this in because when I started my PhD work with Mark Whiteman a long time ago, I started out doing electrochemistry in rat brains, and so we've come full circle back to this, but in flies. And some people have heard me talk about this a few times, but I'm going to give sort of a finished product and a short version of a paper that we have in press that just is about to come out. And then I'm going to talk about a little bit of amperometry detection of exocytosis, small carbon fiber electrodes, from single cells. And so we're going to talk a little bit about neurochemistry. And I'm going to really use this to launch into some experiments that many of you have heard me talk about, but some have not, and in the package, hopefully not. But it's something that I'm very excited about, and very hard experiment, but we've done some beautiful work, and we're working on this to get some measurements of what's inside of single vesicles. These vesicles are only 100, 150 nanometers in diameter, sometimes 50 nanometers in diameter, and we're measuring what's in them one at a time. We're doing that with electrochemistry, so I think it's appropriate. We call it electrochemical cytometry. And then I'll come back to this because we published a paper in the fall in Nature's Scientific Reports on looking at these events and looking at the post-feet, and I'm going to give you that analysis and talk a little bit about that. So it's going to be a little bit of a circle around things. Before I do that, though, I made just a short word slide to thank people. I'm just going to read it because I really feel strongly about this. I feel in awe being up here getting this award. I've watched people get this award back from the beginning. I think I was around for almost, maybe all of them. And I just feel in awe. I don't feel like I deserve it. But I have it, so it's too bad I'm not giving it back. Okay. I wanted to thank all the wonderful students and postdocs I've worked with over the years and still do, along with collaborators and mentors. These people, in my opinion, deserve the lion's share of the credit for anything out of the lab. And I think it's that way all the time. It's the science that is important. Without you, this work would never have happened. So thank you very much. Okay, back to science. In my lab, we're focusing on neurocommunication, no surprise there, and some work on stem cells that I won't talk about. The lab is getting so broad now that I've moved to Sweden in terms of, because of the technologies that we're getting and that we're available to. It's hard to focus in, but here I'm going to try to stay on this track. And we're working with a bunch of different techniques: amperometry, electrochemical cytometry, which I mentioned, in vivo voltammetry, electrochemical imaging, mass spectrometry imaging. I certainly wouldn't talk about that in this group. Get thrown out. Separations. They're allowable nowadays. And capillary electrophoresis with both electrochemical and mass spectrometry detection. Today, as I said, I'm going to talk about electrochemistry in the fly brain, then amperometry, and then electrochemical cytometry. I'll just mention at the end electrochemical imaging, because I don't really have time to go through all of this. Okay, so in vivo measurements and the neurotransmitters in the adult fly. Actually, I took out the larvae. I just realized that. Okay, and we're doing some cool things there too. But I'll mention that for a poster presentation you can go to if you want. This is what the fly looks like in a picture. They look pretty big in these pictures. I'm kind of impressed. They almost look like a big one of those bees that attacks you in one of those monster movies. But they're really tiny. Of course, these are fruit flies. But they're a wonderful model, okay, to look at. And I didn't put in a slide on this, but fruit flies have been studied for a long time for all sorts of models. And there's something like, Karina would correct me if I'm wrong, but there's tens of thousands of mutants that are in a library that you can get. It's a huge number, so I won't quote the number. And they're also famous for the quote that Sarah Palin made during the election a few years ago, where she said that she was going to take money away for a project that she wanted from fruit fly research in Paris, France. And then when they investigated, it turned out that the fruit fly research in Paris, France, was actually a collaboration with some people in Paris with a group at North Carolina, okay, that was working on flies. And the thing she was going to take the money for was autism research. And it turns out that the best non-human model for autism is the fruit fly. It's a particular strain of fruit fly. And it was that fruit fly they were working on. Okay, so, but I tell that story because it tells you, I mean, the range of things. There are Parkinson's flies, there are Alzheimer's flies, there's autistic flies. I mean, they're models for certain aspects of these diseases, and there's a wide range of things you can do. Cell communication, okay, with neurochemistry, and when we start to focus on the fly, here's a blow-up schematic of a fly brain. Neurotransmitters, we know what they are. They're the chemicals that transmit signals from one neuron to another neuron, okay, or at least in the generic sense. And here we have the fly brain. You have the eyes here, and here's the nervous system, if you will. And there's a part called the mushroom body here, and there's a PAM region that has a lot of dopamine neurons in it. Or not a lot, a few dopamine neurons in it, okay? So we can look at dopaminergic neurotransmission in this organism. If you think about it, in this region, you have these neurons that sometimes contain, some of them contain dopamine. And if you focus in on a dopaminergic synapse, what you find is that here's the terminal coming down, and it's going to try to communicate with this terminal. And you have dopamine in these vesicles, and when the signal comes down the neuron, these vesicles are caused to fuse with the plasma membrane at the end of the terminal. Okay, the terminal fiber or the synaptic bouton at the end of this. And they open up and they dump out their transmitter. This is these little balls here. And it goes across and interacts with a receptor. In this case, dopamine is typically after this and actually during this is inactivated in different ways, but part of the inactivation process is taking dopamine back up into the cell. Okay, and this is dopamine transporter called the DAT. Now, most models show it in the synapse. I won't go into this. I mean, in any detail. It's probably not most of them there. But the fact is that you've got this DAT, this protein that takes dopamine back up, and dopamine can be repackaged into the vesicles. This DAT is blocked by cocaine, one of the worst drugs of abuse that are out there. It's also blocked by a drug called methylphenidate. And this is a drug that we've been trying to study a little bit more in detail in terms of its effects relative to cocaine. And cocaine or methylphenidate block dopamine going back into the neuron, leave dopamine out here so it can keep hitting receptors, okay, wherever they are, and across the synapse or on other parts of the cell. And this is how you get part of the euphoric response and so forth with cocaine and methylphenidate, especially cocaine. Cocaine has this structure. Methylphenidate has this structure. Methylphenidate is also called Ritalin. It's a drug that's used for hyperactive children, okay? And so you wonder, giving a drug that does the same thing as cocaine to a hyperactive child, you wonder if this is a smart thing. But there's actually a big difference between cocaine and Ritalin. Cocaine actually is a very fast drug in terms of getting into the brain, causing its action, and then being degraded. All happens in about 20 minutes. Okay? Methylphenidate is much slower crossing the blood-brain barrier, and it takes a while to get to be metabolized. And so it actually is around and has its effect more drawn out over hours. Okay? And so this is a huge difference. This drug is highly, highly addictive, and this is only at best mildly addictive. Okay? And so there's a big difference between the two. If we look at cocaine, lots of piles of cocaine here. Makes it look bad, actually, although I like that. It's on a black surface reflection. Cocaine's a psychoactive drug. It's in the body for about 20 minutes, as I said. It passes the blood-brain barrier fast. It's the second most harmful drug considered by most experts in the field. Second most addictive drug. Again, this is an opinion based from people who treat drug users. There's no effective drug treatment for this drug. A number of people have suggested that because methylphenidate crosses the blood-brain barrier slower and is a slower-acting drug, that we might be able to give this to patients to mitigate or slow down the effects of cocaine. The studies in humans are mixed, but studies with cocaine addiction in humans are hard to do. Okay, and so we need a model system, and that's why we wanted to test it in this. We went and used the fly model. We did fast scan cyclic voltammetry, and I know you're going to talk about that in the next talk all too, so I'm just going to set you up and not talk more in detail on this. But we do fast scan cyclic voltammetry. This audience has heard this over and over again in terms of the basics. We take dopamine, we're looking at dopamine, and I'll talk about the paradigm of how we do this in a minute. And we use carbon fiber cylindrical electrodes, 5 micrometer diameter. We can oxidize dopamine to dopamine orthoquinone and detect its amount. We use color plots. Now, these color plots, I'm not sure who actually started them in your lab. I know Michael Hine did a lot of the programming and stuff for it, so maybe he started. I'm not sure who started them, but I call them Whiteman colors. Okay, there's a reason for that because Mark's red-green colorblind, as many of you at least know, and so these colors are absurd. Okay, okay, but to someone who's red-green colorblind, they're apparently very good. Okay, I'm going to pick on you in advance because I know you're going to pick on me in a few minutes. So, if you look at this, the color plot is a combination of data. We're scanning from minus 0.6 volts to 1 volt to minus 0.6 volts again, and then back. So we're doing cyclic voltammetry, and we do it over and over again across time here. This axis here, the color, is the current. And so green is a high current. Blue is a high oxidation current. Blue is a high reduction current. And these more ugly brown colors are middle currents, okay? They're the baseline, okay? If you plot across, or if you take a chop here, of course, then you get a nice cyclic voltammogram, a current versus voltage plot. And if you take a line across here and then convert this to concentration, you get a concentration versus time plot. And you can see what we've done here is we're actually looking at a change in dopamine concentration from none to up and then down again. And we know it's dopamine because we injected dopamine, and this is a voltammogram for dopamine. Okay. So that's how we do our analysis. This is how we do our quantification, identification, although we usually know what we're looking for in this experiment. Now, the experimental approach for electrochemistry in the fly was developed in collaboration with somebody named Kay Hahn, who's now moved to Texas, El Paso. But it was while I was at Penn State, we started this. And she developed a fly collar. It's this huge thing. This only shows part of it. That's made out of plexiglass with a little millimeter hole in the middle. And what you do is you take the fly, you put it on ice for a little while, and then flies, when they get cold, they're anesthetized. At least they don't move. And you have about 45 seconds to pick them up with a pair of tweezers, stick their head through the hole, and put some agarose on there to hold them in place. Okay, it's a rather fast thing. I'll show you who does this in a minute. Okay, it's a very interesting thing to try to do. I have never done this. I've just watched. Okay, but I can imagine more flies. I complain that the flies get loose all the time. I could just imagine. Okay, then you take a very careful set of micro dissection tools, and you take off the cap on the brain here, and then you can do your experiments. And we treat them different ways that I won't go in detail. Here now, I'll point out, this is the front of the fly here. These are the eyes, so you can see from the direction of the eyes. This is now the front of the head here. And we then take our carbon fiber electrode, and we place it with a micromanipulator into the brain, and a micropipette injector we put down right next to it, right near this, and we can then push into the ejector. We can eject out solution. And in our paradigm, what we do is we eject out dopamine, with what I'll show you today, because dopamine, we're putting out exogenous dopamine, and then we're watching the brain cells that have dopamine uptake transporters pull it away. And we look at the rate that this goes away and the amount that this goes away. And that's how we do this experiment. We use green fluorescent protein labeled brains most of the time. They're labeled, we label specific cells with green fluorescent protein. We actually get these cells out of these flies out of a bank, okay, and sometimes they're given to us. They come from all over. Here now again is the back. Here's where the mushroom bodies are. Our PAM region is right in here. And we use this because this tells us where dopamine neurons are. And so we can go in and identify where to stick this. Okay, that's enough of that. So then when we do this experiment, now remember, Karina Byland, who's sitting in the audience over here, I won't point at her with the laser pointer, but she's done these experiments that I'm going to show you here. A student in my old group, Monique Makos, started out doing a lot of these experiments and did the original setup of this, but Karina's done a lot of beautiful work, just defended her PhD in December. And what we do here is we're now measuring a cross-section. We've got all the data that I won't show you in the color plots from the scans. These are all scanning voltammetry. And we're measuring current versus time. And we're doing this where we've not given any oral methylphenidate to the flies, and also to flies where we've fed them methylphenidate for a few days. Okay? Now, when we feed them methylphenidate, they take it up, and the methylphenidate goes in and blocks the transporter. And so we think what we're testing is whether by blocking that transporter, it will block the then subsequent action of cocaine. Okay, because cocaine we think works at the same transporter. And there's been some arguments about whether they work at different places on the transporter, and so this is an important experiment to look at. When we did this, here is pre-cocaine, and here is post-cocaine. The response goes up significantly, the end gets wider, okay, by a little bit. And so we see that cocaine has an effect of blocking the transporter when we put the dopamine in, because more of it is stuck outside of the cells. Then when we fed methylphenidate, I think this is 15 millimolar methylphenidate for 3 days. When we do this, you can see here is the pre and here's the post, okay? And you can't really look at the absolute levels because it's different based on positioning and so forth, but the difference here is much less here than it is here, okay? Very important. And then I summarize this on the next slide, where we have this plus a lot of other data. This is the summary just of this one data point here, but here is the peak max, the maximum peak normalized, okay, so we normalize through these experiments because of the differences. Then we have time from cocaine incubation, and so this is 10 minutes before we put cocaine on, 5 minutes before, 5 minutes after, etc. Then the color plot, the color scheme here, I'm sorry, Mark, you probably can't see this, but the left side is no methylphenidate. The purple, the next one over, is 5 millimolar methylphenidate. The black is 10 millimolar, the gray is 15, and the last one here is not a methylphenidate at all, but it's something called a fumin mutant. This is a mutant fly that doesn't have the transporter in it. Okay? And so you would expect not to see any difference there, and you really don't. It's basically the same across. Here's the 100% line. The cocaine, you see, makes it go way up, okay, when you don't feed any methylphenidate, and it stays up for a period of time. The flies don't metabolize the cocaine and get rid of it as fast. Then you see here's 5, then here's 10, then here's 15. I like to zero in on any one of these, actually. You can zero in, you can see it drops off pretty nicely, okay, as you go down, although these two are about the same for 0 and 5, and then it kicks in at 5 and 10. So I like this one right here, but you can't tell. Maybe it doesn't have as big of an effect at only 5 millimole. Gives you an idea, though, of what you might try in a human to do this experiment. What range of concentrations? I don't think you'd want to feed a human 5 millimolar, even 5 millimolar methylphenidate for a long time, but you can sort of try to work out relative doses with this kind of an experiment. And so we're pretty interested in this. Now I'm going to stop at this because I have 2 other subjects to talk about, and I don't want to go over time on my own symposium. But for more measurements in Drosophila brain, you can go to Sudi's poster session, which was this morning. Okay, so you can't go to it, but you can find her at the reception. Okay, and you can go tomorrow to Kubra's poster session, which was here. If you're interested in CE electrochemistry of fly brains, and then on Tuesday, tomorrow afternoon, New gives a talk on CEMS of the fly brain for drug addiction studies, very much related to this and part of that paper. Okay, second topic, quantifying vesicular release, okay, at single secretory cells. And that's not the topic, that's the introduction, okay. This is an experiment that was started in Mark Whiteman's lab a long time ago. Boy, it's a long time since you published that paper. But we're doing it on PC12 cells, and we did it originally here, and I put that as well. But we take a carbon fiber microelectrode, place it on a cell. We actually come down and touch it, basically. So we trap a little bit of solution there, stimulate the cell to undergo exocytosis, and measure dopamine release. And I talked about this in the introduction. When we stimulate the cell, as we do here at each one of these arrows, we stimulate a cell over and over again. And we look at the current as a function of time. We get current transients. And if we blow one of these up, they look something like this. And we can integrate the current and get the charge, and then from the Faraday's law, we can get the number of moles that are present in each one of these. And for a number of years, people would ask me, how do you know that this is the amount that's in the vesicle? Especially because my lab in the late 90s went back and back-calculated how many were in the vesicles, how it changed with different treatments and things like this, and we were all wrong, okay? Because it's not everything's coming out, okay? And there's an awful lot of data that suggests this now. And Christian Amantor has been going around saying this from his stuff, and I'll mention that at the end as well. So you have this exocytosis process, and I say, is exocytotic release all or none? And if not, how often and what fraction is released? And so we have this process that comes down, opens up a pore in the membrane as it attaches. Terrible diagram, it would never look like this, but some artistic license. And then, I think, often closes and comes back in again. And I think I've got really good evidence for this now. And I think most of this community, it believes this now. You talk to neuroscientists, and it's not. Okay? I mean, at a recent meeting, Dave Solser came up to me and reminded me again. He said, you know, Andy, realize that most people don't believe you. Okay? This is something you're going to have to convince people. Analytical chemists seem to really understand this. Okay? It is important, okay, because if only a fraction of release of the material in here, the transmitters, is released, then this is important as it could be regulated. And you're probably wondering, I left out, what is the black dot? If you don't know the neurobiology, this is a protein matrix. My picture of a protein matrix is a black dot, okay? And this is also thought to hold transmitter, and it's been hypothesized to do all sorts of things, and it's not really as well understood as people like to think it is. And it does not fill the whole core of this, so that's important as well. So the technique that we've been doing, which was developed in my lab at Penn State by Donna Omiatek, who I think is also in the audience, and is being continued. And also Michael Hine, who's at Arizona, was instrumental in working with Donna and setting up the microfluidic device part of this. We actually do an experiment where we measure vesicle content by separating the vesicles. And this is an electrochemistry audience, so I left out the whole capillary electrophoresis capillary. The end of it is here. And we do capillary electrophoresis into a microfluidic device. We then put in lysis buffer here, and then we run these things against a carbon fiber, again, a cylindrical electrode. As the vesicles elute from the capillary, they come out more or less one at a time. And this is a phenomenon that Edgar Arriaga pioneered some years ago, looking at liposomes and then now very much at mitochondria for many years. And we get these vesicles, but in our device, what we do that's a little different is we use the lysis buffer to focus these vesicles against this carbon fiber electrode. And we think what's happening is that when they come down, come out of here, they come out here, we focus them down, and they hit the carbon fiber electrode, stick and start to lyse, and then we oxidize everything that's in them. Because our models suggest that about 95% of what's in the vesicles is oxidized. 95 plus or minus, and so it's actually about 100% of what's there. And so we think this is what's there, and we're anxious actually to try to test several hypotheses on the mechanism. One thing you have to realize is that when we oxidize these, we get a current transient, looking at current versus time, that is basically the same as we see for exocytosis, but now for a whole vesicle. We can use Faraday's law then to quantify this. And you need to realize that if a vesicle is even just out about here, three-dimensional diffusion will make this go in all directions, and you will see almost nothing up here. And we've done a number of models to show this. You have to be almost the size of the vesicle away from the electrode to have the kind of response that we see. So it basically has to stick, in my mind. We've then turned to PC12 cells. These are these pheochromocytoma cells that we've used a long time to measure exocytosis from. We culture them, we homogenize them. We do a hypoosmotic lyse, which blows up the cell but is supposed to leave the vesicles relatively intact and not lose transmitter from them so much. I don't know how they know that because they couldn't measure it, but this is what most people theorize. Okay, and then we do centrifugation and we pull this off. And we did this for a long time with a kit from Sigma Aldrich, which is no longer available. Okay, we're reinventing things. Okay, so when we do this experiment on the PC12 cells, what we want to do is compare our exocytosis measurements with carbon fiber electrodes, okay, looking at release, okay, so the amperometry, with our cytometry experiments using the microfluidic device and the electrochemistry at the end of the capillary electrophoresis to look at the total contents. And Yan Dong in my lab did these experiments simultaneously with similar cultures, sister cultures, when Donna did these experiments with cytometry. And this is the kind of data that we obtain. And we have here, this is current versus time again, and a blow-up of this region here, you can see for the amperometry, a number of these very sharp peaks. Here, the same thing, current versus time. We blow up a region of these peaks, they look something like this. A little bit of over-filtering causes them to ring down, but otherwise the same. And we can integrate the current, which is, you can see is a little longer here, but it's a different technique. We integrate the current, get the charge, use Faraday's law to find out how many molecules we're looking at. In this case, we're looking at the number of molecules that are released. In this case, the number of molecules that are in the vesicles. When we did this with the PC12 cells, we plotted the number of events over the total number versus the catecholamine amount okay, to the 1/3 power, which is a long story, but zeptomoles, okay, basically. But because it's to the 1/3 power, it gives us a Gaussian representation, roughly Gaussian distribution of radius of the vesicles. And because it's 1/3 power, that there's a big difference, doesn't look so big. Okay, that's the other side of this. This is for release, and this is for the total content. And if you compare the numbers, the number of vesicles looked at, or release events and vesicles in each case, and I'll point out there's a lot of vesicles and a few hours of separation, you get 141 zeptomoles here and 387 zeptomoles here. Those are interesting numbers. I'm going to come back to a number of these things in this last part of the talk in a minute. And this is 85,000 molecules or 220,000 molecules is what we were measuring in those cases. Okay. When we do this, if you ratio, well, first of all, you realize this is high throughput. Okay, that's, if you take that away, it's not as important here. But the average PC12 cell vesicle releases about 40% of its total transmitter load during exocytosis. This is release, this is the amount. That's what we found with this technique. Okay. Now, we also did some experiments with rat vesicles, mouse vesicles. I always want to say rats. And mouse vesicles, we took tissue from mice, pooled them for vesicle isolation, and then looked at them. Very small transients. These vesicles are synaptic vesicles. They're much smaller. And when we did this, and I'll just give you the summary, for about 8,000 vesicles per nanoliter injection volume, we find 55 zeptomoles or 33,000 dopamine molecules in here. Now, this is really important because this is about 0.8 molar for a 50 nanometer vesicle diameter. And all the models out there assume it's about 0.1 molar and that there's at most somewhere between 1 and 10,000 molecules. Okay? That's because all those models are based on release, looking for what's in there, not at the total amount that's in there. We can also do some pharmacology with the mouse. We injected the mouse with reserpine, a drug that takes dopamine out of the vesicles, and L-DOPA, a drug that is used for Parkinson's disease and increases it. And we see a massive increase with a reasonable, maybe a 10 times higher than normal dose of L-DOPA. Not a very high dose. When we give a patient L-DOPA, it's probably doing massive things to their vesicles. And we can do these kinds of experiments. This is sort of an aside to my overall point of my talk. Okay, for more on electrochemical cytometry, Jelena gives a poster. She's the person that's been continuing this work in my group on Wednesday. Wednesday, yes. Okay, so the last thing I want to do is go back to this measurement of exocytosis, look at release, and ask the question, if only some of the materials comes out, does the vesicle then, it must open and close again? Okay? Well, we've been postulating this, and Christian Amator has a beautiful model showing that for the electrochemical events that we have, that you can model it without the vesicle ever opening and get the same response. There's a lot of other data I'll show you, actually, that people haven't really thought about, even some of us who got it, okay, like you and I. And there's a lot of things going on, but what happens, okay, to this? Now, to preface this, I want to talk about an experiment that Lisa Melander and my group did. And we haven't published this, actually, or we sort of have in a new way now we're looking at it. But she did some experiments where she was looking at exocytosis. And when in the old model, I think actually Leslie Chalmers made this diagram or at least modified it at some point. You know, you have the vesicle come up, open up, and people thought there was this rise before you saw the spike. And people thought that this rise was this opening of the fusion pore, and then this was the complete opening to spread out. Okay? Now we don't think this anymore. Now we think that this is actually just a little bit of this is probably smaller, and this size is probably what we're seeing here. Very important to realize that. Now, what happened, what made me really sort of jog me beyond the cytometry, beyond everything else, the cytometry certainly suggested this, okay, that result. But what really pushed me on this was that Lisa did this set of experiments where she incubated cells for, I think, a half an hour or around there with phosphatidylcholine, lysophosphatidylcholine, and phosphatidylethanolamine. And there's a lot of data, a lot of thought and modeling you can put into the slide. But the bottom line is she looked at this time here, the fall time, which if this was opening would be really diffusion, okay, and should be the same for all. And instead it changed. So as we went to PC, it went up. To LPC, it went up even more. PE didn't really change very much. Differences, and we stopped and we said, wait a minute. If the fall time is changing, then there's this clearly closing again. Okay, and so we came up with this idea here, and that's Lisa. You can talk to her at this reception tonight too, because I'll show you that. And so we went looking. We said, well, if there's pre-spike feet, then there should be post-spike feet. And I think several people have sort of said in the past, I think I've seen post-spike feet, and told their research advisors this, and they said, oh, nonsense, or no, don't look at that, okay? And so we went looking, and it was not easy. Lisa will attest to this, but we found them, and you can see some models of these here. The problem with a pre-spike foot the post-spike foot relative to a pre-spike foot is that a pre-spike foot, you can have different shapes. It can be just a long, drawn-out part here, or a different plateau, what we call it, where it flattens out like this. But with a post-spike foot, if it's long, drawn out, it's the same, looks sort of the same as just coming down. Okay? So you can't use all the feet. And there's about 7 or 8 different classifications of these so-called feet for the pre-spike foot. We really have basically one we can use for the post. And that's here. So what we think is happening is the vesicle opens to a fusion pore, opens a little more, then closes again. And if it gets stuck here, you get a little bit dribbling out, okay, here. And this might be more than dribbling. It might be important, actually. Peaks with pre-spike feet, she found, were about 5% of the time. Peaks with post-spike feet, now we've upgraded this for anybody who's seen this before, are 2.3%. And that's because we're only looking at plateaus now. Before we tried to look at everything, and then we got really messed up, and we decided to back off of that. We didn't trust it well enough. So now we really trust this, and Lisa went over it really carefully. I trusted 100.0%. And so here you see, this is the idea here, is this pre- and post-spike foot. Well, now if you get peaks that have both pre- and post-spike foot, if you're losing transmitter here, you've got a reaction vessel, okay? You've got a vessel or a container. And we're dumping a little bit out here, we're dumping a little bit more, a lot more out here, and then we have some leaking out here. Well, the concentration's going to go with the shade here. It goes down. And so the post-spike foot, in the case of where you have 2, which is very rare, looking at plateau feet, but we do have them, okay? The post-spike foot should go down in current, okay? And it does. Now the foot time goes up, so the actual amount released here, on average, goes up with the post-spike foot. But that's because they're longer, seem to be more stable. But the current, the plateau here, is lower, and it's significantly lower. And I'll point out that when you do a lot of fancy calculations, it's about 30% lower, okay? Which is interesting, okay? She also incubated the cells with different phospholipids, lysophosphatidylcholine and phosphatidylethaноlamine. Lysophosphatidylcholine has a bigger head group and a small tail group. Phosphatidylethaноlamine has a small head group and the 2 tail groups. So they're opposite in geometry, and they fit differently in curved membranes differently. Okay? And so this makes a difference in how they'll fit in. Here's something here's a lysophosphatidylcholine would look something like this, and you see it would fit in here, and here's a phosphatidylethaноlamine would fit in here. Phosphatidycholine, which is more cylindrical, that we don't show in any data here, would not fit in well at all in any curvature in a membrane. Okay, so here's control, and we have pre- and post-spike feet, and here is the foot current on this here. Okay? And you can see the current drops down. It drops down even more with LPC, and then much less in terms of the differential with PE. Now, we also looked at the average amount of molecules, and there's a lot of information here. I would run out of time if I tried to explain it. I'm going to run out of time anyway. Okay. And so I won't go into that, but this is interesting because it gives us 3 points on a graph. And if we go through a complicated linear plot, okay, from this, and consider a resistance of the pore as the length over the diffusion coefficient pi times the radius squared, okay, and of this and the time, okay, then what you find is that, or radius, pi r here, okay, and what we find is that we can come up with an alpha, which is the resistance of the pre over the resistance of the post, okay, and we can then back calculate it with the 3 different concentrations to an amount which is about 400,000 molecules is what is in the cell in each vesicle. Okay? Now this is the amount released per event, and it's only using spikes with feet, so it's larger than we would have expected. If we then go back to each individual case, we find that 39% is released from control cells, 48% from LPC-treated cells, 31% for PE-treated cells. And if all events are 39%, because that's what it was for the control, then we go back and look at not just the events that have spikes with feet, but all the events, and we have 290,000 molecules is what we think are in the vesicles on average. It's kind of an interesting calculation to go through. So the evidence for open and closed exocytosis, I'm out of time, so I'm going to just buzz through this. But the mismatch between release and cytometry data I talked about, mismatch in release and content in mammalian cells, and the time for amperometric event closing is affected by the specific link of incubation. And the post-spike feed are critical here, is looking for this evidence. But I'll point out that there's a lot of other things that I'm happy to talk about afterwards. QCM experiments, release change with osmotic pressure and temperature and things like this. Christy Haynes and Leslie Sombers did experiments before in Mark's and my groups. And flickering fusion pore with Solser's group. Patch amperometry leads to more coming out because they suck it out, okay, with the patch. Models of diffusion that Christian has developed are beautiful now, showing this. And then even patch clamp and turf and other things are probably consistent with this if you think about it long enough, okay? And that I'll leave you with that. And so what I think happens is that you have you might have kiss and run, which has been described before, but I call this extended kiss and run, and I think this is what we normally look at with amperometry. Okay? I think this is what we normally see. I think we see these about 2% of the time, and I won't go into why, but we look for large events. It turns out that our computer programs, our data analysis programs, cut those out because they were too long and too large. Okay, so people weren't looking at them. And this is very interesting, and you can find them. For more on this, Lisa talked about this on Sunday. Again, you can find her at the reception. She's really a wealth of information on this particular set of experiments. We've fabricated microelectrode arrays following Bo's work, and we've got up to 36 electrodes now. And this is just an advertisement because all these people are doing things with this. Johan, you can find at the reception. Raphael, unfortunately, is opposite this symposium, but you can find him at the reception. And Jun is the one who has a lot of data on this I didn't have time to talk about. And with that, I'm going to just thank the audience and thank you for allowing me to blatantly advertise the other talks. And thank you, SEAC, for the award. And thank you, the committee, for losing your mind when you decided to do it. And I want to thank my group, the people with the halos. I think they're all here. No, Lynn is back in China now, but they're all here. They did the work that I talked about. And I want to thank the funding agencies, and I want to thank you for your attention. 2 minutes over.
Thank you, Andy. We have time for one or two quick questions. And we only have one microphone, so. Mark?
I was wondering, Andy, in your analysis of brain vesicles, could your number possibly be low? Because when you isolate the preparation and prepare them, they have a chance for leakage out of the vesicles.
In all the...
actually be higher than what you're saying.
That's right. And usually when I'm not rushing through things, I say that. You're right. There's no question. I mean, if there's an error in any particular direction, the odds are it'll be in too low, not too high. It's very difficult to imagine a situation, at least so far, where you'd have buildup of these during the preparation or the isolation protocol. It's more likely that we lose some, and so it might be actually that there's 50,000 in there instead of 33. The hypoosmotic lyse is supposed to prevent leakage from the vesicles, but you know how that is. I'm not sure I believe it. So. Okay.
Any other questions for Andy? Well, congratulations again.